Imagine a tiny microscopic nightclub operating right inside your cells. And the mitochondria, right, that's the VIP power plant generating all the energy.
Right, the engine room.
Exactly. So standing right at the velvet rope of this VIP section is this massive, heavily armored bouncer, and he's controlling exactly who gets in and out. But what if cancer cells figured out a way to bribe this bouncer?
Yeah, to essentially hack the system.
Right. Not just to keep the cell's power plant running nonstop, but to actively lock out the immune system's top assassins, like allowing the cancer to just live forever. And I mean, that biological loophole, it isn't science fiction. It's the centerpiece of this groundbreaking 2026 multi-omics study published in partnership with the Hormel Institute and Ningbo University. So our mission for today's deep dive is to unpack the newly decoded secrets of this microscopic bouncer, which is a protein called VDAC2.
And we are looking at a really massive pan-cancer approach here. They're mapping the overarching strategies that malignant cells use to survive. So this isn't just about understanding one localized tumor type. It's about exposing a structural vulnerability in cancer's armor across the board.
Right. But before we look at how this spreads across the whole body, I feel like we need to examine the engine room of a single cell first. So let's look at VDAC2, or voltage-dependent anion channel 2. I mean, it's a pore-forming protein sitting right on the outer mitochondrial membrane.
Yeah, and in a healthy cell, its day job is pretty straightforward. It's shuttling metabolites like ATP out into the cell and bringing ADP in. So it regulates the energy flow.
Okay, but when a cell becomes malignant, what exactly is VDAC2 doing that makes it so critical to the tumor's survival? Like, what's the bribe?
Well, it's basically hijacking the fundamental metabolism of the cell because, cancer relies heavily on the Warburg effect.
Right, that metabolic shift where it burns glucose incredibly fast without needing oxygen.
Exactly. And VDAC2 is the channel that directly facilitates this heightened glycolytic flex. The tumor aggressively upregulates VDAC2, which essentially jams the mitochondrial gates wide open to keep that twisted engine running at a chaotic speed. It just rapidly fuels the biosynthesis required for a tumor to multiply.
Wait, hold on. If VDAC2 forces the cell to burn glucose at this hyperfast anaerobic rate, wouldn't that massive energy spike eventually just, burn the cell out? Because anaerobic glycolysis generates a ton of toxic byproducts and cellular stress.
Oh, absolutely. It's highly toxic.
So how does the cancer cell sustain that level of metabolic chaos without triggering its own destruction?
And that is exactly where VDAC2 performs its second and honestly perhaps more insidious function. So we know that when a cell sustains massive DNA damage or metabolic stress, the BAK protein normally triggers apoptosis.
Apoptosis being the programmed cell death, right?
Yeah.
The self-destruct button.
Right. And BAK does this by oligomerizing, basically clumping together to puncture holes in the mitochondria. That releases cytochrome C and initiates the cell's self-destruct cascade. But VDAC2 actually physically binds to the inactive form of BAK at the mitochondrial membrane, and it just sequesters it.
Wow. So it's not just blocking a chemical signal, it's like physically handcuffing the executioner.
Precisely. It holds BAK hostage in its inactive state. Because VDA2 prevents those BK proteins from clumping together and forming those lethal pores, the cell simply cannot pull the self-destruct switch, no matter how much metabolic stress it's under.
So just to do a quick recap here for you listening to lock this in, the cancer cell is shoveling coal into the furnace at a dangerous speed to feed itself, while simultaneously dismantling the exact protein designed to shut the furnace down. That is an incredibly potent survival advantage.
It really is the ultimate immortality switch.
But okay, let's unpack this a bit more. If this physical binding is happening at such a microscopic, localized level, how do researchers know this isn't just some weird biological quirk? I mean, how do we know this isn't just an artifact of a few specific cell lines they grew in a petri dish?
Yeah, that's a very fair question. And to prove this was a universal mechanism, the researchers pulled back from the microscope and employed a really massive big data approach. They used a three-pronged multi-omics methodology.
Okay, break that down for us.
Sure. So they used bulk RNA sequencing, single-cell RNA sequencing, and spatial transcriptomics. And they analyzed thousands of patient samples across these massive public tumor atlases like TCGA and GTX.
Right, and the methodology here is really crucial to understand the scale of this finding. So for those of you listening, think of it like trying to understand the ecosystem of a massive fruit orchard.
I like that analogy, yeah.
So bulk RNA sequencing is like taking all the fruit from the orchard, throwing it into a giant blender, and testing the smoothie to see what genetic ingredients are present. You get a broad average, right? Then, single-cell RNA sequencing allows you to separate that smoothie back out, and examine the individual pieces of fruit one by one.
Exactly, which captures the immense heterogeneity of the tumor. You see all the different cell types.
But then you have spatial transcriptomics, which is the really cool part.
Oh, it's revolutionary. Taking that orchard analogy further, spatial transcriptomics is like having a precise GPS map showing exactly where every single piece of fruit was growing.
Like on which specific branch next to which other fruits.
Yes, it preserves the geographic context of the RNA expression. And by applying that GPS mapping, alongside some heavy machine learning algorithms, they found something staggering. VDLT2 wasn't an isolated anomaly at all.
So it's everywhere.
It was heavily upregulated, meaning its production was just an overdrive in the vast majority of pan cancers.
Wow. So it really is a universal defense mechanism.
It is. But the spatial transcriptomics gave them the most critical insight. Because they had the geographical layout, they saw that the cells overproducing VDAC2 weren't just randomly scattered throughout the tissue.
Where were they?
They were heavily concentrated right in the active, aggressive tumor margins, the outer edges. And most importantly, these VDAC2 overexpressing cells were physically intertwined with the body's immune cells.
Wait, I'm trying to wrap my head around that dynamic, because if the tumor is sitting right there, physically interacting with the immune system, why aren't the killer T cells wiping it out?
Well,
I mean, the immune system isn't blind. If a cluster of cells goes rogue, starts hoarding energy, and shuts down its apoptosis pathways, massive chemical alarms should be going off, right? The T cells should be swarming that exact geographic location.
and that's the thing, the T cells are swarming and the chemical alarms are going off.
They are.
Yes. And this brings us to the core discovery of the study. It's called the VDAC2-AK1 IFN gamma axis. The immune system's primary assassins, which are the CD8 positive T cells, they are doing exactly what they're programmed to do.
Okay, so what goes wrong?
When they spot these malignant cells, they release interferon gamma, or IFN gamma. And you can think of IFN gamma as a highly specific chemical launch code.
A launch code. Okay, I like that visual.
Yeah, it binds to receptors on the cancer cell.
Yeah.
And that initiates a signaling cascade that is supposed to tell that BAK protein to detonate and destroy the mitochondria.
Right. The launch code goes to the bomb. Boom, cancer cell is gone. That's the normal biological protocol for clearing out malignant cells.
But the tumors are overproducing VDAC2 precisely to absorb that impact.
Oh, wow.
Because the VDAC2 proteins are so heavily concentrated at the mitochondrial membrane, they physically intercept that signaling cascade. They maintain their iron grip on beak, and they completely block the IFN gamma signals from ever activating it.
That is wild. So the tumor is actively using VDAC2 as an immunosuppressive cloaking device.
Exactly. It effectively acts as a shield.
That completely changes the paradigm of tumor evasion. I mean, the tumor isn't just hiding from the T cells in some dark corner of the body. It's standing right in front of them, catching the launch codes midair and neutralizing them before they can trigger the explosives.
And because the T cells are constantly firing these signals and getting absolutely no response, the surrounding microenvironment becomes highly immunosuppressive. The data actually showed a significant drop in the infiltration of active CD8 positive T cells into the deeper tumor microenvironment over time.
So the immune system essentially exhausts itself trying to break through and then just gets locked out of the area completely.
Yes, exactly.
But to prove this wasn't just a statistical correlation in the big data, the researchers tested this dynamic in real time in the lab, right?
They did, yeah. They utilized in vitro gastric cancer models, specifically focusing on a highly aggressive cell line called AGS. When researchers took these VDAC2 overexpressing gastric cancer cells and co-cultured them directly with active CD8 positive T cells, the cancer cells didn't just survive the encounter.
What did they do?
They actively proliferated and invaded further into the surrounding matrix. And furthermore, the researchers measured a drastic, quantifiable drop in the protective IFN gamma levels in that culture.
Wow. So the cancer was actively silencing the immune system's primary weapon. So time for another quick mini recap for you all. The tumor isn't just hiding, it's actively catching and neutralizing the immune system's launch codes. It disarms your killer T cells to create a safe haven. But if the tumor has this perfect, impenetrable defense at the mitochondrial level, how do we even begin to break through it?
Well, that's the million-dollar question in oncology right now.
Because if VDAC2 is the villain shield, we can't just keep throwing T cells at it. We have to figure out who is giving the genetic orders to build the shield in the first place, right?
Exactly. To map out that chain of command, the researchers turned to specialized databases that track cellular transcription networks. They were hunting for the upstream modulators.
The upstream regulators, so the specific genetic managers that tell the cell to manufacture VDAC2.
Right. And they uncovered two main types of control mechanisms at play here, starting with transcription factors.
Okay, let's visualize these transcription factors carefully because the physical mechanism really matters here. Think of them as complex chemical keys that have to fit perfectly into the ignition of the VDAC2 gene to start the manufacturing process.
That's a great way to put it. So the researchers identified a specific transcription factor called PTTG1 that acts as the master ignition key. PTTG1 is actually an oncogene itself, and it's normally involved in regulating cell division.
But here it's been hijacked.
Completely hijacked. When PTTG1 binds to the promoter region of the VDAC2 gene, it cranks the engine, flooding the cell with these bouncers. But conversely, they found another factor called NFATC2 that acts as a repressor.
So a different key that tries to shut the engine down.
Right, but it's often totally overpowered in these malignant environments.
Okay, so the tumor is somehow keeping the PTTG1 key constantly turned in the ignition. But the study didn't stop at transcription factors, did it? Because they also looked at epigenetics and physical RNA splicing errors, which just adds a whole new layer of complexity.
It does. Epigenetics plays a massive role here.
Since we know cancer relies heavily on epigenetic modification to silence tumor suppressor genes, how exactly is it manipulating methylation to control VDAC2?
Well, interestingly, in this specific pathway, it's actually about the lack of silencing. So they looked at promoter methylation at the CPG islands.
And for context, these are the dense chemical tags that cells normally use to tightly coil up a gene and prevent it from being read by RNA polymerase.
Exactly. But in colorectal cancer models, they found that the level of these silencing tags inversely correlated with VDAC2 expression. The tumor had actively stripped away the normal epigenetic locks, leaving the gene wide open for continuous, unchecked transcription by PTTG1.
Unbelievable. It systematically removes the structural safety mechanisms. And what about the splicing error they found? Because the methodology here used some really specialized sequencing tools to look at how the RNA blueprint is physically cut and pasted together after it's transcribed.
Right. So when a cell reads a gene, it has to splice the RNA together. It cuts out the non-coding introns and stitches together the exons, almost like editing a film strip.
Okay, follow.
The researchers discovered a structural mechanical glitch in this process. It's an alternate promoter splicing event specifically located at a region called exon 2.
Ah, so it's like a mechanical glitch in the cell's photocopier. In that specific exon 2 region, the cellular machinery skips a crucial line of code.
Yeah, and that exact typo doesn't break the protein, which is the crazy part. It actually forces the cell to print out a highly stable, hyperactive variant of VDAC2 that might be even better at binding BA.
That is terrifying.
It is. And that structural typo leads directly to the massive overproduction and enhanced survival capabilities we see driving the disease. Understanding this network, the PTTG1 activation, the epigenetic unlocking, and the exon 2 splicing is critical because it completely redefines our therapeutic strategy.
Right, because if a pharmaceutical drug can't crack the physical VDAC2 shield at the mitochondria, maybe because it's too structurally tough or deeply embedded in the membrane, we don't attack the shield directly.
Exactly. We dismantle the supply chain.
We design a drug that specifically blocks the PTTG1 chemical key from entering the ignition. Or we find a way to target that exon 2 splicing glitch to correct the photocopier typo from the inside out, long before the shield ever gets built.
That is molecular sabotage at its finest.
It really is. But let's bring this from the realm of molecular theory down to the reality of the clinic, right? How does mapping all of this actually change patient care? I mean, if we know exactly how the cell misreads the VDC2 blueprint, could we theoretically test a patient to see if that misreading is already happening? Is that what the gastric cancer screening data was looking at?
Yes, absolutely. The diagnostic potential is one of the most immediate, real-world applications of this data. The study highlighted the potential of VDAC2 as a biomarker, and they calculated a receiver operating characteristic curve, an ROC curve, specifically for gastric cancer.
Okay, so to evaluate how reliable a biomarker is, we look at the area under the curve, or AUC. For you listening, an AUC of 0.5 means the test is essentially a coin toss. It's useless. But an AUC of 1.0 is mathematically perfect.
Right. And when they tested VDAC2 expression as a diagnostic indicator, the AUC was 0.808.
Wow, 0.808. To put that in perspective, that means if you take a random patient with gastric cancer and a random healthy patient, this test will correctly identify the cancer patient over 80% of the time based solely on their VDAC2 levels.
And coupled with an over 86% sensitivity rate, that makes it an exceptionally powerful screening tool. You could theoretically test for this specific upregulation and catch the cancer building its defenses long before the tumor really progresses.
That's incredible. But diagnostics provide the early warning. Therapeutics are what alter the outcome. So to figure out how to chemically attack this pathway, the researchers utilized a computational algorithm called XSUM, right?
They did. They ran the VDAC2 expression profiles against these massive pharmaceutical databases like CMAP and GDSC that catalog exactly how thousands of different human cancer cell lines respond to various chemical compounds.
So they basically unleashed an AI algorithm to find the exact chemical lockpicks for the VDAC2 shield. What did the data reveal?
Well, initially it confirmed what oncologists already observe in the clinic, unfortunately. Tumors with high VDAC2 levels are stubbornly resistant to a wide array of standard chemotherapies.
So the shield works beautifully against blunt force, highly toxic attacks.
It does. However, the algorithm identified specific targeted compounds drugs designated as C2ATKN by 35874, MS275, and W by 13 that are highly effective against VDAT2 expressing cells.
Okay, wait. How did those specific drugs bypass the resistance when standard chemotherapy fails?
While the exact binding mechanics are still being mapped out, the pharmacological profiling suggests these compounds exploit the specific metabolic dependencies created by VDAC2. So, for example, MS275 is known as an HDAC inhibitor.
Okay, HDACs are enzymes that normally wind DNA tighter around histones, right?
Yes. And MS275 interferes with that epigenetic machinery. Remember how we discussed the tumor stripping away those chemical methylation tags to keep VDAC2 turned on?
Yeah, the CPG islands.
Right. MS275 effectively disrupts that epigenetic control. It alters the way the DNA is spooled, forcing the cell to alter its gene expression and potentially collapsing the VDAC2 supply chain entirely.
So instead of trying to batter down the mitochondrial door, the drug alters the cellular environment so the door can't even function. That's brilliant. And this brings us to what I think is the most profound clinical takeaway of the entire study. The data shows that high VDAC2 expression generally leads to a very poor prognosis for the patient. But crucially, that poor prognosis can be partially reversed if the patient naturally possesses a high infiltration of CD8 positive T cells.
And this is the critical intersection of targeted therapy and modern immunotherapy. Right now, immunotherapy using checkpoint inhibitors to supercharge the patient's own T cells that is the frontier of oncology.
But as we've established, immunotherapy fails if the tumor has a VDC2 shield absorbing all the T cell signaling.
Exactly. So the strategy becomes a highly coordinated strike.
Tell me more about that.
If we use these newly identified compounds like MS275 to knock out the VDAC2 expression first, we strip away the tumor's cloaking device. We remove the protein that's handcuffing BAC. And by doing that, we leave the tumor completely exposed to the immune system. When the T cells release their IFN gamma launch codes this time, there is nothing left to absorb the impact.
The BA protein is finally free to oligomerize, it detonates the mitochondria, and the cancer cell dies.
You knock out the bouncer, and then you let the immune system raid the club.
I love that. It transforms VDAC2 from just an observational biomarker into a highly actionable therapeutic target. It's a tool that predicts standard drug failure, a diagnostic marker for early detection, and the exact vulnerability we need to exploit to unlock the full potential of immunotherapy.
It bridges the critical gap between understanding the molecular mechanism of a disease and weaponizing that understanding to fundamentally alter the course of treatment.
It's just phenomenal. So taking a step back to look at the massive journey we've been on today, we started with a microscopic gatekeeper on the mitochondria, controlling hyperfast metabolism and stopping programmed cell death. We explored how spatial transcriptomics mapped this gatekeeper across the entire pan-cancer landscape, revealing how it physically intercepts our immune system's lethal signals. And finally, we saw how AI is decoding the epigenetic networks and splicing glitches to find the exact chemical inhibitors that will shut this gatekeeper down.
This research really reinforces a vital shift in how we approach oncology. For decades, the focus was primarily on what makes cancer grow the accelerated engine of cell division. But this study proves that to truly defeat these malignancies, we have to understand how cancer actively manipulates its environment to survive.
It's not just a passive lump of cells.
No, cancer isn't just a fast-growing mass of chaotic cells. It is a highly adaptive entity that engineers complex structural defenses into its own biology. By mapping vulnerabilities like VDA2, we aren't just slowing the cancer down, We are turning its own survival mechanisms into its ultimate downfall.
It completely changes how you view the microscopic world operating inside of you every single second. Which leaves me with a final thought for you to mull over as you go about your day. If a tumor goes to such incredible, complex lengths to build this intricate VAC2 shield around its cellular power plants, what other everyday metabolic processes might it be hiding in plain sight? Could the very energy systems that make cancer so aggressive today also be the exact keys to its complete undoing tomorrow? Take a look at your own energy habits today, the fuel you give your body, and wonder what's happening at your cellular gates right now. Thanks for joining us on this deep dive.